This next recording is going to look at the formed elements of blood. We first are going to look at the red blood cells, also called erythrocytes. Notice that erythrocytes are a component of the formed elements, and we count their quantities in millions. We find 4.6 to 6.2 millions per microliter in males, to 4.2 to 5.4 million per microliter in females. You should have an idea of the numbers of the millions per microliter of red blood cells in males and females. Red blood cells are biconcave cells. They're flexible discs, and the purpose of the shape is to provide an increased surface area for packaging more hemoglobin. These are very special cells in that when mature, these cells lack a nucleus and they lack organelles. They are essentially a little structure whose job is to carry hemoglobin. So we're looking at these tiny little cells that are filled with hemoglobin. You can see here their biconcave shape. They look like a cough drop, and that shape increases the surface area of the cell, allowing for more oxygen to reach the surface of the cell. The cells have no nucleus or organelles. They're flexible because they have to travel within very, very tiny blood vessels. The tiniest of blood vessels are called capillaries, and these blood vessels are so small that cells have to travel single file. So they need to be flexible to be able to fit into these very tiny capillaries. The cells are filled with a protein called hemoglobin that has the ability to capture oxygen. So the function of hemoglobin is transport of oxygen from lungs to tissues. Some of the carbon dioxide made by tissues is also captured by hemoglobin and transported back to the lungs, is captured in the tissues, the carbon dioxide, and transported back to the lungs. When blood is hemolyzed, when the red blood cells burst, we call that hemolysis of blood. And when that happens, the red blood cells burst and blood becomes translucent. So you can see here the first two tubes are opaque because they have intact red blood cells, whereas the third tube looks translucent because the cells in it have been ruptured or hemolyzed. So we mentioned that red blood cells are filled with a protein called hemoglobin. This is a globular protein, meaning it has a circular shape. And the hemoglobin consists of two chains of polypeptides. Two are called alpha chains and two are called beta chains. Each chain is bound to a group called the heme group, a chemical group called the heme group. The heme group in turn contains one iron atom, which is the atom that actually physically binds to oxygen. So you should know the structure of hemoglobin. We're going to see here in this PowerPoint the two orange color peptides are going to be the beta ones here and here. The red ones are the alpha globulins. Each one of these two betas and two alphas have the heme group, which is this the blue circle in the middle of the molecules. This is a representation of a heme group. Notice the iron in the middle, which is the molecule the atom that actually attaches to oxygen. And what's surrounding the iron is the heme group, which is made of rings of carbon with nitrogens attached. So these structure here are carbons bound to hydrogens and attached to nitrogens. But the part of the molecule that actually binds to oxygen is the iron. Notice that each hemoglobin has 4 heme groups, 1 per globin. Each heme group has 1 iron, which means that a hemoglobin molecule can bind a maximum of 4 oxygens. Embryonic and fetal hemoglobin must be able to capture oxygen under the low oxygen environments found in the uterus, which means that embryonic hemoglobin and fetal hemoglobin have a higher affinity for oxygen than what we call adult hemoglobin, which is the hemoglobin that appears after birth. So fetal hemoglobin quickly changes into adult hemoglobin the minute the baby takes in its first breath. Iron is the main component of the heme group, is the atom that actually is going to capture oxygen. Atom is a precious element in the living world. It is very hard to absorb. We humans absorb iron in the upper GI tract. Vitamin D typically helps in the absorption of iron. Remember that we find 1 iron atom in a heme group. Each binds 1 oxygen. There are 4 heme groups per hemoglobin, which means that each hemoglobin molecule can bind a maximum of 4 oxygens. Hemoglobin, when it is bound to oxygen, the molecule turns a bright red color called oxyhemoglobin. Hemoglobin without oxygen has a dull color called deoxyhemoglobin, and it has a dark red shade to it. Hemoglobin bound to carbon dioxide is called carbaminohemoglobin, and it also has a dark red color to it. So the bright red blood that is found in arteries is oxyhemoglobin, which is hemoglobin bound to oxygen. Hemoglobin bound, the blood found in veins, it's deoxy and carbaminohemoglobin, so it has a dark red color. Notice the difference in this PowerPoint between the darker red of deoxy and carbaminohemoglobin and the bright red of oxyhemoglobin. Notably, when hemoglobin binds to carbon monoxide, which is a poison, it turns a bright red color, which makes it harder to detect the poison. The patient will not have the typical signs of someone who is depleted of oxygen, which will be a bluish coloration in the skin. Instead, the person may look healthy because of the bright red color of the hemoglobin bound to the carbon monoxide. Now, the process of making red blood cells is called erythropoiesis. And as we mentioned in past PowerPoints, it takes place inside red bone marrow. Hemocytoblasts are the germ cells from which all blood cells come about. Some of the hemocytoblasts will mature into proerythroblasts, from which red blood cells are going to arise. The process of making red blood cells is called erythropoiesis, and it takes about 4 days. As these proerythroblasts mature, they are going to turn into erythroblasts. From there, into reticulocytes, as these cells lose their nucleus and their organelles and begin to accumulate hemoglobin. The final circulating erythrocyte is going to be the mature cell that can actually carry hemoglobin. Anytime you see the term blast in the name of a cell, that means a young cell. So this is a picture of blood. you can see the red globules are blood cells, but notice that some of the blood cells are healthy in that, or normal, I would say, in that they look empty inside, which is how they should look. However, some of them look like they have stuff inside of them. You can see the granules. Those are reticulocytes that were let out of circulation, probably due to anemia. So if there aren't enough red blood cells, red bone marrow will begin to allow for young red blood cells like reticulocytes to leave the marrow and start circulating, even though they are not ready yet. This one right here is even a younger cell. Here you can really see the nucleus inside the cell. This is a nucleated red blood cell, extremely young. It should never have been let out into circulation, but there's probably an anemia, a severe anemia going on, and that's why the red marrow is allowing for these not yet mature cells to be let out into circulation. It is normal to find a small number of reticulocytes in blood. 0.25 to 2.5% of the red blood cells in the blood may be reticulocytes, and that's normal. However, anything higher than that is definitely a sign of anemia. Finding nucleated red blood cells in blood is never normal, and it's definitely a sign of severe anemia. One of the nutrients that are needed for erythropoiesis, which again is the process that makes red blood cells, are folate and vitamin B12, and folate is another form of vitamin B. These vitamins are needed for making DNA. So any cell that is rapidly replicating and maturing needs to be making DNA, and that's why vitamin B is needed for the formation of red blood cells. Obviously, iron is also needed. Low blood oxygen levels in the environment and anemia will lead to a slowdown in erythropoiesis. When that happens, when that happens, a hormone is called erythropoietin. So erythropoietin, or EPO, is a hormone secreted by the kidneys under low oxygen environments or under anemic conditions. Erythropoietin, or EPO, has two functions. It stimulates the production of proerythroblasts by red bone marrow, and it accelerates red blood cell maturation. Both of these events are going to end up increasing the numbers of red blood cells in blood. So ultimately, erythropoietin is going to increase red blood cell production by stimulating red bone marrow into producing young proerythroblasts, and also by accelerating the maturation of red blood cells, which, remember, they take about 4 days from beginning to end. So you should know what erythropoietin is. You should know that the organ that secretes erythropoietin are the kidneys, and what the function of erythropoietin is. Red blood cells live for about 120 days. All red blood cells are destroyed in the spleen. So remember, they are made in the red bone marrow. They take about 4 days to be matured. They're put into circulation. Once in circulation, they live for about 120 days. And as they get older, they become less flexible. So as they travel through the spleen, and the spleen tissue is like a net that these blood cells, these red blood cells, have to navigate. The older they get, the less flexible they are, so they're going to get caught in the net, in the tissue network of fibers, protein fibers that are found in the spleen. The spleen is made by reticular connective tissue. So it has this net of protein fibers that trap the old red blood cells. Once they're trapped, phagocytes, macrophages, white blood cells, immune system cells, large immune system cells called macrophages, are going to go to these cells that are trapped and begin to eat them up, to phagocytize them. This destruction of red blood cells occurs all the time. Over 2 million red blood cells are destroyed in the spleen every second, meaning that out of the 25 trillion red blood cells in circulation at any time, 2.5 million are being destroyed per second, which means that 2.5 million are going to have to be replaced every second. So about 1% of the total red blood cells in the body are replaced each day, which is tremendous, and it speaks of the tremendous job that the red marrow is doing, constantly making red blood cells. Once the red blood cell is destroyed, the hemoglobin inside is going to be disassembled. Now, remember we said iron is a precious element in the living world. Iron is never discarded as a waste product. Instead, the cell is going to recycle and reuse the iron. So the hemoglobin is disassembled into the globin part, which are the amino acids, the polypeptides. Then from there, we get the heme separated from the globin. The heme was the ring of carbons with nitrogen, and the iron in the middle of the heme is removed and is going to be recycled and reused. The heme without the iron is called biliverdin, and from biliverdin, which is put into circulation, is then called bilirubin. Free bilirubin is converted to conjugated bilirubin by the liver. Bilirubin is a toxic substance. It has a yellow color to it, and the body needs to eliminate it. The body is going to eliminate bilirubin through the bile in the large intestine, and it's going to be taken out of the body through feces. Some of it also travels through the kidneys and is going to be eliminated in urine. So the yellow coloration is in urine is the bilirubin that is being eliminated. So just review this cycle of what happens to red blood cells after they are destroyed. So the hemoglobin is broken down. The globin part, which are the polypeptides, the amino acids, the protein part, if you will, is going to be disassembled into individual amino acids that can be reused. What's left is the heme group. From the heme group, the iron is going to be recycled. So the iron is going to be taken into the liver, where it's going to be stored and eventually transported to the bone marrow to be made into more hemoglobin. So notice that iron is stored in the liver and the spleen for that matter also. The heme group without the iron is called biliverdin, which then is converted to bilirubin. Free bilirubin is the bilirubin found in the blood that is transported into the liver, and the liver is going to conjugate it and convert it into eliminated through the bile. Some of the bilirubin is also going to travel to the kidneys, where it's going to be eliminated in the form of urine. So you should know the fate of hemoglobin once the red blood cells is destroyed. You should know what bilirubin is, where it comes from, and how it's eliminated. When there are abnormal conditions with the liver, let's say hepatitis or cirrhosis of the liver or liver cancer, it cannot process the bilirubin and it remains in circulation. Remember, this is a yellow substance. So as it travels through the blood, it's going to cause what we call jaundice. Jaundice is a yellow coloration of the skin and the white of the eyes due to the accumulation of bilirubin in the blood. The causes are, again, problems with the liver, infections blockage. There could be a blockage in the pancreas also, and the connection between pancreas and liver is obstructed, and so bilirubin accumulates and goes into the blood. It could be an obstruction of the bile ducts. It could also be an excessive destruction of red blood cells, but that is something that happens typically in babies. So jaundice due to an excessive destruction of red blood cells is a problem with babies that may have an incompatibility of blood with the mother. And that's what sometimes babies are born yellow. So you should know what jaundice is and what is it an indication of in adults. So you should know the characteristics of a mature erythrocyte. What do they carry inside? What is hemoglobin? What is the structure of hemoglobin? And what does it bind? Know the characteristics of a mature erythrocyte and the reasons why they lose their nucleus and their organelles. I mean, their function is to carry hemoglobin, so they need to empty themselves of everything else so that they have more room for the hemoglobin. Down here it says a blood smear of an anemic patient shows many reticulocytes and nucleated red blood cells. What is the significance of that? So remember, reticulocytes and nucleated red blood cells should never be found in large concentrations in blood, especially nucleated red blood cells should never be found in blood. Pernicious anemia, we haven't really talked about it. Pernicious anemia is a problem with vitamin B, is the inability of the patient to absorb vitamin B. This leads to iron deficiency anemia. Oh, I'm sorry. Pernicious anemia leads to anemia because the patient doesn't have vitamin B to make hemoglobin. And then why would an iron deficiency diet lead to anemia? It's because you don't have the iron for the hemoglobin. You should know what EPO is, which organ secretes EPO, and what is the specific role of EPO in erythropoiesis. Which would be to increase erythroblast production and increase the maturation of red blood cells. Blood doping, we didn't really talk about that. Sometimes athletes may take in EPO, erythropoietin, in order to make more red blood cells, giving them an advantage because now they can carry more oxygen. Or they may even just get a blood transfusion to get more red blood cells. You should know the average lifespan of an erythrocyte and also where are destroyed, and the components of hemoglobin once they're broken down. And again, you should know what jaundice is and what kinds of conditions cause jaundice.